Thank you. To shoot my time, I just go to the two things, two viruses and visualize models. Yeah, Kerr and Frazier has mentioned the herpesviruses. Now, I'm not going to go to the one by one by that, but I'm going to mention some disease burden. So, we know it's not congenital infection, we have the TORCH. What's the TORCH? Toxoplasma, rubella, what are the congenital infection pathogens? When I say TORCH is discarded by the Chinese doctor, why? Because this detection cannot tell the doctor if the baby is infected or not. So it's very hard for them to say, oh, the baby, you need to terminate it or carry on. No, it's very hard because they cannot, the doctors have 90 patients a day. There's very limited time for the doctors to go through all of the data. So, and another thing is like in the adult, 1/3 of the adult have shingles. And also the organ transplant recipient, tumor patient, a lot of patients, a lot of the people have probably is the final herpes infection will be the final straw to break them down. For example, tumor patient, in our memory, in our knowledge, should be like the tumor, multiple tumor grow big and bigger, and the patient should be skinny and bony, right? That's the way it should be the tumor patient to die of. Unfortunately, it's not that situation. When they did the chemo or radiologic therapy, then suddenly the patient just gone. So, in my hand, it's like, for example, the pneumonia patient from different samples. It shows ABV, CMV, and HSV, a lot of samples. That's pretty high there. That gives me a hint. Maybe something worse to re-evaluate what's going on there. For example, one case, VZV infection. One day, the hospital guy, the expert, the eye doctor, phoned me. It's like, oh, Min-hua, we need your help. Then I say, what happened? A 42-year-old lady have two eyes, both eyes, acute retinal necrosis. That means maybe both eyes will branch. Then they treated with ganciclovir, avacinclovir. They treated it, but still the eyes goes worse and worse. Then I told them, yeah, use the brevidine to treat it. After 3-week treatment and 2 weeks, wait 2 weeks to review, check the eyes, it's come back. By this way, we saved the lady's 2 eyes, and they keep pretty good eyesight. Another case, CMV infection, congenital CMV infection, even the IgG is positive. For example, China has a pretty high CMV infection, over 90%. And one of the lady had 20 weeks. Ultrasound shows the placenta has problem, lung has problem, heart has problem, and also the MRI shows the placenta and the brain has problem. And also after the termination, they do the MRI and CT shows the brain has problem and the lung has problem. And we also showed the HE staining and the CMV antigen IE1, IE2 staining. It's typical CMV infection. It's like they have nuclear inclusion body, giant cells there, and also for the calcification, it shows like Vancouver or something. Yeah, classifications there. Case 3. We met a patient, AIDS patient. Yeah, they have severe immune deficiency and also have severe lung problem. And then the lung tissue and the non-tissue same way staining, and also the COVID stain. These big cells actually is this same way cell. So the same way is there. Then I would say we should reconsider the herpes virus burden. Usually we'll say another herpes, who cares, right? Actually, maybe we really need to think about it. So, like the infection features, yeah, primary infection, latency, reactivation. That's a feature. And another thing, biologic feature, herpes virus is a big guy. The smallest one is 125 kb. The genome is large and complicated. And also, if we need to do something to manipulate it, yeah, we have to have the back system or other system to manipulate. But half of these genes is non-essential. But how non-essential? Because this will be regulate the latency and reactivation. But if we have these things, we have the technology or platform to manipulate the virus. We will have more ways to do it. For example, we design the virus, make the infection visible. That will be way more easy for us to study the virus host interaction and the neuropathy. And by this, since we have manipulated, we can make the neural tracers, viral tracers, or better vaccine, or the vectors. So by this, I would say we have HSV1, 2, VZV, CMV, and MCV, they back to manipulate it. I would like to say here, I need to thank Tom and Waju for those reagent for the support. So part 1, I'm going to mention a little bit about the HSV1. The HSV1 has a strain H129. This virus has very special character. They just the primary goes from upstream, up neuron to downstream neuron. So we call it integrate once, spread. Then how we will mention the tracers we made, 2 viruses we made, and how we're going to talk a little bit about how the virus do the external transport, how the transsynaptic spread. The first one, out of the HSV H129, the genome, we just add 4 GFP. When we just add 4 GFP, the virus is so bright. Then we just inject the virus to the mouse brain. A few days later, we know the virus spread all over the brain. Then how we do it? We do it in F-most. That means the whole brain, we don't slice it. We just connect the information, influence message from that, a signal from it. Then once we connected this layer, then we erase the layer so that the whole brain, we can have the whole brain like the huge, big book. Then if we want to take one of the page out, we just look at it, then we can identify how these neurons connect to each other, different layers. So we call it like multi-polysynaptic tracer. It's like this virus go to this neuron, they can go to the downstream neuron and further downstream neuron. further downstream neurons. So we call it polysynaptic. And for the neuroscience, say, if we get the tools from A to B to C, it's very hard for us to identify, say, that's from A to B and to C. Maybe it came from the A directly to C. So we need to get rid of one of the essential genes to make this virus can go to walk one step. For example, we get rid of the GK gene. GK is the late gene. So we get rid of this, then we use AAV to complementary this TK. Once these two viruses go to this same cell, then this virus, HSV, lack of the TK gene, they can replicate in this neuron, then go down one step. And here, since there's no TK here, so they cannot walk further. So they call it monosynaptic. When we get this AAV to the brain, then after 3 weeks, the TK gene accumulates enough. Then we get the deficient, GK deficient virus here, we call it tracer. Then wait a few days, then we can see if this virus from the initial site to go down. So we got from the OB to different area, brain area. So from here, actually it's like we have DTK, right? And then we make DGK. Why we make DGK? Because DGK the virus genome can replicate. Then they can amplify the signal. And once the lack of TK, this virus cannot replicate in the neuron. So the signal is weak, but the GK is much more stronger. So by this, when we get GK here, the downstream neuron is very bright labeled. So we get this new tracers. But the problem is, we needed to get to the AAV first, then 3 weeks later, we get another virus to the same spot. We hope to the same spot. But 3 weeks later, you try to make sure the virus go to the same spot, go to the same cell. It's very difficult. And we need to wait for finish this experiment, need one month. Can we do something, make something new tracers to make it easier? For example, if we can get 2 viruses injected at the same time, then we don't have to the space mismatch. Just get to the cells, get to the spot we want. So we make the applicant, H129 applicant. Then we'll get the applicant and we'll have the helper, HS helper. These 2 viruses have the same character. They covered, packed with the same HSV protein. So they say once the character is the same, then we can mix these viruses here, and then we can, a few days later, we're going to check. So after we check that, it's 1 day, 24 hours later, we know it's brightly labeled as a neuron. Then 3 days or 5 days later, the downstream neuron is also labeled. So, and this neuron, the morphology of this is very bright, and the morphology keep well, even the biophysiology keep normal. So then we know we made the new tracers. Yeah. That is faster. We just need single injection and fast and brighter for the labeling. So then the question we need to answer is, when we claim this virus is integrate from upstream to downstream, you need to know how the virus transport in the exon. Once it go to the end, how they cross from the one neuron to the next one. So that's two questions we need to answer. To answer this question, we have to make the two systems. It's like, first, microfluidic culture system. We need to culture the neuron, and the neuron has to be mature and connect to each other, form the synapse. Then we need to have the two viruses. The two viruses means you need to see the virus, how they move. So we made these two viruses. So we now, when we get the outer layer, have the green coat, and the inner layer, its capsid, have the green jacket. Then we say, okay, if then we get infected, watch the virus particles go transport in the exon. Then later we count them. If the virus just transport with the capsid or with the intact neuron, intact variant, so based on that, we count it, and we know, okay, at the beginning, they move slower, and later they move fast, and most of the particle is capsid, not the entire virus. Then we can make another two. We just make the virus just have the one color, the capsid have the red one. Make the neuron green, then you know that the virus just goes to the spikes. Then I say, okay, if that's on the spikes, can they go through the synapse? Yes, the virus just crossed the synapse. The questions followed. It's how they cross synapse. Like this, fusion together make a channel, or transcytosis. How does that? So make another two. Make the GD fuse with the pH lowering. Once this pH lowering will fuse with the GD, when this virus in the cell, the pH is lower, then the fluorines is very low. Once they release out to the cell, they're brighter. So we know, yeah, from the presynaptic neuron, yeah, these viruses were released. Then follow questions. Does this associate with the neural activity? Yeah. When we use the potassium chloride to activate the neuron, make it excited, the virus just released. But in the controls, yeah, they are there. Yeah, different time, different stage when they release, it's there. And calcium is very important for the neural excitement. So the calcium inflow, it's like when we use the stop vector to stop the calcium inflow, then the virus just stuck there. And when we collect the calcium with the EGTA, yeah, virus also stuck there. So that means it's really associated with the calcium. And also we've identified which molecule to sensate and release it. So I'm not going into the details. So now we have different polymolysynaptic traces. Now it's used by the neuroscientists all over the world, and we know how the virus transport and they cross the synapse. Another one is CMV. Felicia has mentioned that. It's like pretty much my time is working on the CMV. And what I'm doing, I'm doing is the congenital infection. So it's like, how can we make it clear to, I don't know why it's okay. I can't move to the next one. Oh. How come I cannot go to the next one?
So, Luo, I would like to remind you that time is limited.
Just tiny.
How long is going to be? How long is the part 2 going to be?
Yeah. So the virus, the congenital infection is the most common birth defect, the most common cause of it. So it's like we lack of models. We're making the models. Congenital infection because that's the major target cell in the fetal brain. So, and this cell is the key for brain development. But the cell is the cell. The cell cannot show the brain structure and the function. So in the whole thing, it's like everybody, when we go to the mouse model, we try to show the truly brain structure and the neural function. At that time, everybody just get the virus to the brain or get to the belly and started the hearing loss. Why they started the hair loss? Because hair loss, same way infection causes the hair loss. That's the most frequent disease in the. But if we want to do the animal model, we have same way just infect human and human cells. Then we have to use the MCMV. So we optimize these conditions and get the virus to the baby. And now it's like we know the infection causes smaller growth retardation and also brain infection and brain damage. And also the hearing loss. Well, we can see the virus, but we really needed to make the virus brighter to say, okay, can visibly follow, tracking the virus infection. By the GFP, we can see the virus. But when you see, you cannot see it very clearly because that's just a bunch of it's like a pile of the ferns. We need to make like this whole brain and the single neuron level and also the MR level to really track the infection and the brain damage. So we do using the MR system to tracking the brain damage and also the different situation of the neurofunctional. So we have this cell model and the mouse model. It's non-lethal, long-term follow-up model. So we are creating more tools and hopefully a better model to help people to understand what's happening there, to get early diagnosis, really the brain damage and the treatment. Thank you.
Very much. Maybe questions? Okay, I guess my audiences are really looking for dinner.
It's kind of too late.
Thank you very much for this wonderful talk. Okay, here we are closing our session and also the talk of today. Let's thank all the speakers of today and especially the session that I chaired. Have a good evening.